All right, there is a request for a quick video on how ribosomes work in translation. So I'm going to do that one right now for y'all. So we're going to start out with just some mRNA and we're going to assemble the ribosome on it in initiation. So here is our mRNA molecule, mRNA. On this end it has a poly A tail, on this end it has a 5' cap. Now we're going to assemble onto this our first tRNA, and the first tRNA here is going to hold methionine, and it's going to be a start site tRNA, so it's going to recognize the sequence AUG. So that means that it's going to have the complementary sequence of UAC on its anticodon. So here's its anticodon, and here is its methionine that it's holding on to. Methionine is just an amino acid. Okay. So now what's going to happen next is we're going to have the small subunit of the ribosome assemble on top of this. Okay. The small subunit of the ribosome right here. And this tRNA, and only this tRNA, is going to come in right in the middle of this. All of the rest of the tRNAs are going to come in on this side of the small subunit of the ribosome. Once this part has assembled, now the large subunit of the ribosome can assemble on top of this, and it's going to assemble up here on this part. There we go. That's not so bad. So here's the large subunit of the ribosome. And the large subunit is going to have 3 spots in it that a tRNA can fit in. So this spot here is called the peptidyl site. It's also going to have a site called the amino acyl site and a site called the exit site. So we have in the middle the peptidyl site, also called the P site, the A site, and the exit site. So 3 sites. Usually, like I mentioned before, the tRNAs are going to come in on the amino acyl site. Then the ribosome will translocate and they will go into the peptidyl site and then finally into the exit site. But the first tRNA that comes in holding onto that methionine is going to come in right in the middle. So that's bound to AUG. Once we have a tRNA in the peptidyl site, now we're going to get a tRNA in the A site, in the amino acyl site. So in this case, we're going to have the sequence here of CUU. And I'll just put in more sequence down here, the next codon. So we'll have a tRNA that reads the codon CUU. And this tRNA is going to have the sequence GAA. And it's going to be connected to the amino acid leucine. Okay. The next thing that's going to happen is a peptide bond is going to form between methionine and leucine. The tRNA that is in the A site is going to be the tRNA that will hold the chain of amino acids, the peptide chain. So the methionine will become disattached from its tRNA and form a peptide bond with the leucine. When this happens, that is going to cause the ribosome to translocate down 3 nucleotides, 1 codon, over to the downstream end of the mRNA molecule. Okay. So here is our piece of mRNA. We still have our AUGs over here. Forgot to translocate it. Our CUU is here, and now our AAA has moved over here. As this tRNA goes into the exit site, that exit site disrupts the pair bonding between the anticodon and the codon, and so this naked tRNA, it's lost its methionine, is just going to exit. So our UAC tRNA is going to exit the ribosome. That's the exit site. Once we've had this translocation occur, now this tRNA is going to be in this site. It stays bound to that CUU. That's still our GAA tRNA, GAA. Only now it's holding on to its leucine that it came with, as well as the methionine that formed a peptide bond to the leucine. Now we have room here in the A site, the amino acid site, for a new tRNA. So this tRNA, lysine holding tRNA, is going to come in with a UUU anticodon holding onto lysine right here. There's our lysine amino acid. And the next thing that's going to happen is that this leucine is going to form a peptide bond with the lysine to make a longer chain. So the methionine was encoded by the first codon, the leucine was encoded by the second codon, and the lysine was encoded by the third codon. And in this way, we're going to get a string of amino acids that are going to match the string of nucleotides. Quick reminder, exit site, no tRNA can bind there. The tRNA exits as it moves into the exit site. P site, the peptidal, the peptide chain carrying tRNA goes there. Oh, I made a big blot. Okay. E site, P site, A site, the amino acid tRNA goes here. The amino acid tRNA is holding a single amino acid.